Hello, welcome to part 1 of your lecture on sleep and biological rhythms. This is Dr. Sally Serafin. In this lecture, we're going to be addressing the question of what is sleep. In the process, we'll cover the stages of sleep, what brain activity looks like during sleep, and answer the age-old question of why do we sleep in the first place. In part 2 of this Sleep and Biological Rhythms lecture, we will discuss circadian rhythms, Zeitgebers or timekeepers, and also explore disorders of sleep. Over the course of the night, we cycle through 5 different stages of sleep, and it takes about 90 minutes for us to complete each cycle. Non-REM or non-rapid eye movement sleep happens in stages 1 through 4, and REM sleep happens in stage 5 and is accompanied by vivid dreams as well as quick eye movements. You will recall from your introductory psychology course lab work that EEG or electroencephalography is used to study brain function by monitoring signals through the scalp and the skull. Well, this is also applied to the study of sleep, in which case electrodes can allow researchers to measure electrophysiological activity from the brain and muscles in the face and around the eyes that signal or indicate particular phases of sleep. So in the case of the electrodes on the scalp, they monitor brain activity, and the two basic types or patterns of activity that we're looking for that help differentiate sleep from or rest from non-rest include alpha activity, which is most marked when resting, and beta activity, which is more marked when we are alert, and it's often noted by some level of desynchronization. So EEG helps us to record the particular patterns of waveform activity corresponding to different phases of sleep. In these two images you see labeled A and B. We have, in the case of A, synchronous delta activity, and in the case of B, desynchronous beta wave activity. In the case of A, if the cells are active all, and by cells I mean neurons, all at the same time, their electrical messages are synchronized and appear as these large rhythmic EEG waves, synchronous delta activity. And in the case of B, neurons are active at different times, and their electrical messages are desynchronized and appear as small chaotic waveforms without any clear EEG pattern. So now that we have reintroduced the concept of EEG and brain waves, you can see that each stage of sleep has a corresponding level of brain activity that can be used to identify it, that is its signature form or waveform. So with waking, we have alpha between 8 and 12 hertz and beta between 13 and 30 hertz in terms of the characteristic signals. You would also expect some desynchronization as different brain areas are doing different things. With stage 1 or non-REM sleep 1, there's a transition between sleep and wakefulness. The theta waves are evident here, and people will experience hypnic jerks that last, this period lasts about 10 minutes. In stage 2, Non-REM, there's theta activity again. Sleep spindles, K complexes, lasts about 15 minutes. In stage 3, we have slow wave sleep, which is high amplitude delta activity sleep. It's the deepest stage of sleep, and it lasts about 1 hour and is again non-REM sleep. REM stage sleep or stage R sleep is often marked by an abrupt change. The EEG becomes desynchronized. There's rapid eye movement. There is the experience of paralysis. People will experience dreaming and can be easily awakened by meaningful stimuli. As you can see from this illustration, sleep alternates between periods of REM and non-REM, each cycle lasting about 90 minutes and containing 20 to 30-minute periods of REM sleep. Slow wave sleep occurs during the first 3 hours of the night. Later in the night, there are periods of non-REM and mostly stage 2 sleep, and periods of REM sleep become longer, as indicated by the larger widths of the bars. As indicated in your reading, REM sleep is marked by EEG desynchronization or rapid and irregular waves, a lack of muscle tone or semi-paralysis, rapid eye movements, and dreaming, for instance, among other things. It's important to note that dreams occur at other stages of sleep as well as in REM sleep. REM dreams are somewhat different than non-REM dreams. Non-REM dreams tend to be shorter, more thought-like, more repetitive, and concerned with daily tasks. By contrast, the kinds of dreams we have when we are experiencing rapid eye movement sleep tend to be less logical, more prone to plot shifts, and can be biologically significant in terms of their themes. Several brain areas are important in terms of their functioning in different sleep phases. The prefrontal cortex, the extrastriate cortex, and the striate cortex are highlighted in this illustration from your textbook. It's important to note that during REM sleep and when we're dreaming, cerebral blood flow is high in the extrastriate or visual cortex, which kind of makes sense because you're having some visual imagery in your dreams. And it's lower in the striate or primary visual cortex and prefrontal cortex. Brain activity in certain areas is also associated with lucid dreaming. The eye movements may be related to visual imagery in dreams when we are experiencing REM and dreaming. The brain mechanisms that are active during dreaming in REM are the same as those that would be active in real life, where we're acting out the things that we're dreaming. In the case of slow wave sleep, brain activity can accompany dreamlike imagery. It's localized in terms of cerebral blood flow in visual and auditory cortices. As you've probably noted from reading the textbook, a lot of what we know about sleep actually comes from disease models of sleep or sleeplessness, such as insomnia. Those of you who watched the movie Insomnia as instructed for this course will note that the lack of sleep leads to a number of deficits or adverse effects that map onto problems with thinking, increased likelihood of illness, and increased susceptibility to death. So the increased likelihood of illness is morbidity, the increased likelihood of death is mortality, and thinking obviously is cognition. So cognition is disrupted by sleep deprivation, especially when it comes to tasks that require attention or vigilance. Extended sleep loss also can lead to adverse health outcomes, increases in type 2 diabetes and obesity and hypertension. Um, and it's really difficult to regain sleep that is once lost. And the effects of sleep deprivation can be fatal. This has been demonstrated in studies of rats, and in particular in the sad case of the inherited neurological disorder, fatal familial insomnia, which results in damage to parts of the thalamus and can lead to death after 12 months because sleep essentially has the life preserving function of destroying free radicals that otherwise accumulate and lead to damaging effects on the brain. While physical exercise is helpful in terms of promoting better rest during sleep, the relationship between sleep and exercise is kind of subtle, and sleep is certainly not needed for the restoration of physical functioning or physiological functioning. The effect of cognitive activity on slow wave sleep has been studied, and it's thought that um, uh, cognitive activity enables the brain to rest and recover from daily cognitive activity in the form of slow wave sleep, which increases after a day or weeks of intense cerebral activity. So, although the functions of REM sleep are thought to persist throughout the lifespan, it is definitely the case that younger individuals experience more REM sleep than older individuals. In fact, the level of REM sleep declines significantly from infancy and childhood to later life. And it's thought that deficiencies in REM sleep can in some cases be made up later. This is what's known as the rebound phenomenon. But as you can imagine, with chronic sleep deprivation, the ability really to rebound and recover all lost sleep hours is greatly diminished. Um, we know that REM sleep helps with the consolidation of long-term memory, and in particular that it facilitates the consolidation of non-declarative memories, whereas slow wave sleep facilitates consolidation of declarative memories, things that are facts. So the brain rehearses newly learned information during slow wave sleep. This is why slow wave sleep may be particularly important for college students like yourselves. In a series of studies um, by Mednick et al. That are described in your textbook, it was shown that only after 90 minutes of napping, both slow wave sleep and REM sleep um, contributed to an improvement in participants' performance on a non-declarative visual discrimination task. And these illustrations pulled from your textbook further illustrate this point, showing that people who stayed awake scored 29, and those who had a nap scored 45, and so on. If you have not already done so, I encourage you to check out the Pearson video on research on college students and how this impacts sleep and learning. So now we're going to turn our attention to thinking about some of the physiological mechanisms of sleep and waking, particularly focusing on the neural control of sleep, arousal, sleep-waking transitions, and the transition to REM in particular. You will recall from the psychopharmacology lecture that sleep is partly regulated by the accumulation of adenosine in the brain. So the brain produces this sleep-promoting substance that accumulates during wakeful hours and is then destroyed during sleep. Adenosine is a nucleoside neuromodulator that promotes sleep. If wakefulness is prolonged, adenosine accumulates and it inhibits neural activity, causing us to feel drowsy. And genetic factors are thought to affect the duration of slow wave sleep in conjunction with adenosine, as noted in your textbook. Here is another illustration from the movie Insomnia that you watched. It's important for you to remember 5 factors in addition to adenosine that are associated with sleep and wakefulness. In the case of arousal or wakefulness, there are circuits of neurons that secrete different neurotransmitters that play an important role in our level of alertness or wakefulness, which is arousal. And they include acetylcholine, norepinephrine, serotonin, histamine, and orexin. As you can see from this textbook illustration, the levels of acetylcholine are higher in both the neocortex and the hippocampus during wakefulness and also in REM sleep relative to slow wave sleep. So acetylcholine is a key neurotransmitter that is associated with arousal, and it is active in both of these brain regions. Norepinephrine, which is the same as noradrenaline, is adrenaline or epinephrine that's secreted in the brain and is associated with waking, and it's a catecholamine. It's part of that class of neurotransmitters called catecholamines, and as a catecholamine, any agonists that influence its levels produce arousal or sleeplessness. And this kind of makes sense because noradrenaline is part of the fight or flight response. And so you would expect it to be lower during stages of sleep like slow wave sleep and even REM sleep, and higher during periods of wakefulness. Another neurotransmitter, in this case a monoamine, that is associated with sleep-wake cycles is serotonin, which plays a role in activating behavior. Almost all of the brain's serotonergic neurons are in the raphe nuclei, and stimulation there causes locomotion and cortical arousal, so it's most active during waking, as you can see from this graph, where it begins to decline essentially from the time that you were drowsy through the different stages of sleep. Histamine and orexin follow a similar pattern in that they are lowest during slow wave and REM sleep and higher when we are awake. In the case of histamine, histaminergic neurons, the neurons that use histamine, are located in the tuberomammillary nucleus of the hypothalamus. They are directly related to cortical activation and arousal, similar to serotonin, which we mentioned before, and indirectly they increase the levels of acetylcholine, so they're higher when we're awake. Orexin projects to almost every part of the brain and has an excitatory influence, so it's high when we're alert, active, waking, especially when we're engaged in exploratory behavior. And the drug that's used to treat narcolepsy, modafinil, actually produces alerting effects by stimulating the release of orexin in the brain. When you get a chance, I'd like you to closely examine Table 9.3 from your textbook, which summarizes each of the 5 or 6 chemicals, so the 5 neurotransmitters and then adenosine, the neuromodulator, that all affect our level of sleepiness or sleepfulness and wakefulness, and make a note of where they are in the brain and whether their levels are high or low in different stages of sleep. So in this lecture, we have talked about the 5 stages of sleep, brain activity in terms of characteristic brain waves in different stages of sleep, and neurotransmitter, neuromodulator levels in the different stages of sleep. And we've also touched on the reasons why we sleep, how it benefits our health and cognitive functions. So in the next lecture, we'll be reviewing some other material related to circadian rhythms and disorders of sleeping.